This is Lab 8 Carbohydrate Catabolism, Part 2 of Cellular Respiration. The first experiment we conducted had to do with cellular respiration, and our goal was to measure the respiration rate of germinating and ungerminating peas by measuring the amount of CO2 they produce. The question that we had to answer was which set of peas will demonstrate the highest rate of cellular respiration. So our hypothesis was the set of germinated peas will demonstrate the highest rate of cellular respiration because they require energy for rapid cell growth in order to grow into plants. So the first part of our procedure was assembly and incubation. So first we located 2 bottles filled with germinated and ungerminated peas. Then we fit the rubber stoppers with the attached glass tubes into the respiration bottles. After that, we submerged the rubber tubing into the water-filled test tubes at our workstation. And then we inserted the rubber stoppers into each glass tube and took note of the time. And after that, we incubated each bottle for 60 minutes. This is part 2 of our procedure, so now we're moving on to measuring the rate of respiration. So after the 60 minutes was up, we replaced the water in each test tube with phenol red. And then we resubmerged the rubber tubing below the surface of the liquid, of the phenol red. After that, we removed the rubber stopper from the top of each jar and slowly poured water into each jar. So the carbonic acid was displaced and it moved through the rubber tubing into the tube filled with phenol red. So an indication of that carbonic acid was present would be a yellow color. So if we saw yellow bubbles forming, we would know that carbonic acid would be present, or was present. So basically what we're doing with this experiment is preventing gas dispersion. So the glucose that would be oxidized from within the peas would produce CO2 or carbon dioxide, and we would trap that within the glass apparatus containing the rubber stoppers. And after that, when we would add the water in step 2, over here, when we would pour water into each jar, the addition of water to CO2, to that CO2 that was trapped, would result or produce carbonic acid, which we would then measure with the phenol red, and the carbonic acid would also be trapped within the tubes because they are sealed and so it cannot escape. So that's what we're doing. We're preventing the gas dispersion so we can ultimately measure the rate of respiration. The second experiment that we did alongside our original cellular respiration experiment has to do with enzymes. So for the enzyme experiment, we had to determine the effect of enzymes on the rate of respiration. So our hypothesis was that the peas will not be able to conduct cellular respiration after they are boiled because their enzymes would have denatured due to the high temperature. This is our experimental design for our enzyme experiment. So first we poured a bottle of germinated peas into a beaker of boiling water and we let the peas boil for 5 minutes. After the 5 minutes was up, we then emptied the boiling water from the beaker and poured room temperature water onto the boiled peas. So after they cooled off, we poured the peas back into the bottle. And after that, we added the experimental apparatus, which was the glass tubing with the rubber tube, etc., to the bottle and added the rubber stopper to it. After that, we placed the plastic tubing into the test tube filled with water, and then we let it incubate for 60 minutes alongside our cellular respiration bottles with germinated and ungerminated peas. Anyway, after that, after the 60 minutes was up, we replaced the water in the test tube with phenol red and resubmerged the rubber tubing. Then we removed the rubber stopper from the bottle, slowly poured water into it, and measured the amount of carbonic acid that was displaced, again seen by a yellow color. And then the less carbonic acid displaced, the more probability that the enzymes had become denatured. Here's a photo of our setup before, or just as soon as we started the 60-minute incubation. So we have the germinated bottle of peas right here, our germinated boiled peas right here, and our ungerminated peas right over here. And you can see Dr. Weber giving us an approving look at our experiment so far. Only 27 seconds into the experiment. So here's a video of Bobby pouring the water to the germinated peas and phenol red, and we can see over here that yes, yellow bubbles and a yellow color is appearing. So what does this mean? This means that carbonic acid is being displaced. This video shows us adding water to the germinated boiled peas, and we can see that the phenol, yes, it's bubbling, and there are bubbles, but there is no color change. All we see is red, there is no apparent yellow. So what does that mean? Again, no carbonic acid is shown here, but yes, in the germinated peas, there was carbonic acid because it turned yellow. But germinated boiled, nope, no carbonic acid. Lastly, this is us pouring water into our ungerminated peas and phenol. We can see that, yes, again, there are bubbles, very similar to the germinated boiled peas, but no, there is no apparent yellow, there is no signs of yellow. So what does this mean? No carbonic acid was displaced. So let's quickly go over our results again. So for the germinated peas, carbonic acid was displaced because yellow bubbles were produced with the addition of water. Germinated boiled peas, no carbonic acid was displaced because no yellow bubbles were produced with the addition of water, and the same with ungerminated peas. No carbonic acid was displaced because no bubbles that were yellow were produced after the addition of water. So lastly and most importantly, we have come to our conclusion, and our conclusion for germinated peas is that they demonstrate the highest rate of cellular respiration because the most carbonic acid was displaced. For the germinated boiled peas, we have concluded that they do not demonstrate any rate of cellular respiration because the enzyme within its cells were denatured when they were boiled. And our conclusion for ungerminated peas is that they also do not demonstrate any rate of cellular respiration because ungerminated peas are dormant and cannot grow into plants. So they have no need for energy and to make energy. However, germinated peas do have the need to make energy and produce energy. That is why they have the highest rate of cellular respiration.